Hi, everybody. Welcome back to biology for our second week of distance learning. This is Miss P coming back with DNA replication this week. So last week we talked about the structure of DNA itself. We learned all about the different molecules that make up DNA, and we learned a little bit about the structure of DNA itself. You guys made a model of DNA, either a drawing or a 3D model using origami. You learned about the people who found DNA and discovered it, and you had a couple great conversations about DNA itself and also about how this whole distance learning thing is going to work. You guys are off to a really great start. I'm super proud of the work you've been putting in, and I'm really excited for you guys to learn about our topic this week. I've heard you guys make questions about this in the past, about DNA replication. We talked about it during the cell cycle briefly. We talked about when DNA copies itself, and you guys were always asking me, oh, how does that work? How does DNA actually copy itself? How does it divide? This week, we're going to learn the answers to those questions. Along with that major question, what is DNA replication in the first place? I have some other big questions for you guys to consider this week. One of them is, how does DNA copy itself? What happens to the original DNA when the copy gets made? And do different organisms copy their DNA differently? These are great questions to keep in mind as we go through the video, and they're also a great base of questions to come back to once you've finished the video and once you've worked on this material a little bit this week and done a couple of the other learning tasks. Come back to these questions. Do you feel comfortable answering them? They're a great way for you to gauge, for you to test, for you to tell if you are on track with this material or if you need some extra help, some extra practice perhaps. Along with these big questions, I always like to have learning goals for you guys. So this week we have 2 learning goals, and these are the big things, the big ideas that I want you guys to take away from this week of online learning. The first one is that you should be able to explain or show how DNA is replicated by the end of the week. You should understand that process well enough to be able to explain it to me or to show me how it works. Another goal that I want you guys to be able to meet by the end of the week is that you should be able to describe how DNA replication is different in different kinds of organisms. I gave the example of eukaryotes and prokaryotes, but I would just like you to be able to describe how DNA replication is different in different kinds of organisms. So with these goals in mind, make sure you have a piece of paper, something to write with, something to take notes on, because we are going to get started very shortly. All right, this week I thought I'd have a little bit of fun with you guys while we are learning, so I included a few science funnies, if you will. I like them. I think that they are just the coolest thing ever when I can take something that is science related and make it a little bit funny, a little bit relatable to some things that we may have common experiences with. So maybe take a look at these if you find time to this week. I think that they are just the bee's knees. So to start out our conversation about how DNA is replicated, I want you to think back to last week. Do you remember what DNA looks like? So on the screen right now, I have a few things that we are going to think about when we think about DNA. The first thing I want you to think about is this nucleotide. So last week we talked about there being 3 pieces that make up DNA. 3 molecules that go into a DNA molecule that make it DNA. So we know that we have a phosphate group, a sugar, and a nitrogen base. So this phosphate group and the sugar combine and they make up what is called the sugar phosphate backbone. So when we look at our coiled DNA, our double helix over here, this major structure for DNA, this yellow band on the outside is the sugar phosphate backbone. The other part of nucleotides are those nitrogen bases. So we have 4 nitrogen bases in DNA. They are adenine, thymine, cytosine, and guanine. And they are the actual code portion of DNA. They are the thing about DNA that makes all of us who we are. depending on the pattern of nitrogen bases your DNA has, you will have different traits and characteristics, and you will look different than other organisms. All right, my first check-in question for you guys this week during this video is right here. My question for you is, when is DNA copied during the cell cycle? I have a picture of the cell cycle right here to kind of give you a little bit of a reminder. It's been a little bit since we've talked about the cell cycle, but I know you guys understood it pretty well when we covered it earlier this semester, and I want to see what you guys remember. So this is a check-in for just you. So take a minute and jot down your answer to that question. When is DNA copied during the cell cycle? You can write this right on your notes. This is for you to test your knowledge to see how much you remember. All right, welcome back. I hope you have thought about this question about when DNA is copied during the cell cycle. So we remember that there are 4 major parts to the cell cycle. We have these 3, G1, S, and G2, that make up a phase called interphase. And then we also have M phase, or mitosis, for the most part. This is the phase where the cell divides. So the M could stand for mitosis or meiosis. We've talked about both of those processes. Both of those are types of cell division. And I want to know when DNA is copied in this process. In the cell's life cycle, when is DNA copied? When is it replicated? Over here is a little sheet to remind us of kind of what's going on in the cell at different parts of the cycle. So as we can see, G1 or gap 1 is mainly growth. S phase is where we actually see DNA being synthesized. So synthesis is a word that means to make more. So if we're making more DNA, that means this is where DNA is being copied. So our answer to our question, DNA is copied during synthesis. Do you want to take a minute and review the other parts of the cell cycle? Please feel free to do so. This won't be on our test. It won't make its way into our test, but it's a good review, nonetheless, of stuff that we've already covered. So what is the best way to copy DNA? Is it as simple as our bodies putting DNA into a copy machine and that copy machine spitting out more copies of DNA? What is the actual copy machine in our cells that makes these vast quantities of DNA all of the time? Because remember, during mitosis, every one of those new cells gets another copy of DNA. So DNA has to be copied hundreds of thousands of times every day. How do our bodies manage that? We must have one efficient copy machine, that's for sure. So, for the purpose of our unit and for this week, I want you to focus on DNA replication in terms of steps and the enzymes that do those steps. So enzymes are protein molecules that do work in our cells and our bodies. And we're going to talk about 3 specific enzymes that really help out DNA replication. And we'll cover them during this process, during the video. But we're going to talk about them in regards to each of the 3 steps. So at each of these steps, there's going to be an enzyme that goes with that. So as you're taking notes, pay attention to the step of DNA replication, but also pay attention to what's happening during that step. So to begin, DNA replication has 3 steps. We have a step called unwinding and unzipping. We have a step called base pairing. And we have a step called joining. And there are different enzymes active at each of these portions of DNA replication. This is an image of DNA being replicated. By the end of today, you should be able to explain this image to me. Remember our learning goal that we are able to explain or show how DNA is replicated. This image would be one great image to study and to be able to maybe draw, to be able to explain. to meet that learning goal. So the first step of DNA replication is called unwinding and unzipping. So remember, DNA is our genetic code, right? So this code is that pattern of nitrogen bases that make up the inside of our DNA double helix. They're found on the insides. They're protected by that sugar phosphate backbone. So in order for anything to get inside our DNA to be able to copy that code on the inside, we need to be able to get there. So we have to unwind and unzip our DNA. If it helps, you can think about DNA as being like a zipper. And we need this zipper piece right here to help us unwind and unzip our DNA. This is going to be played by an enzyme. The role of this will be played by an enzyme in our bodies. That enzyme's name is called DNA helicase. So on this picture, this purple enzyme right here is called helicase. It is the enzyme responsible for unzipping and unwinding our DNA. So it attaches to a DNA molecule and it unwinds that double helix and separates those nitrogen bases that always pair together so that in the future, another enzyme can attach to these strands of DNA and make copies. So remember, those enzymes are those protein molecules that do work in cells. And for the first step, our enzyme is called helicase. As it unwinds that DNA, a replication fork is formed on the DNA. So here we have our helicase enzyme. It's unwinding the DNA, moving in this direction. And as it unwinds, a replication fork forms behind it. So think about a fork in a road or a fork in your path. You're going to have two options. There's going to be two separated strands. This area right here is the replication fork. That was step 1 of DNA replication. Now we are moving on to step 2, base pairing. So now the DNA helicase has done its job. It has unwound and unzipped our DNA and formed this replication fork. We now have 2 single strands of DNA. In this picture, they are the blue strands, and we're going to call these strands parent strands. These parent strands are going to act as a template to add new nucleotides to as we make copies of our DNA. So as we add these new nucleotides, we still have to follow those same pairing patterns or pairing rules that we learned last week. So here's a spot for you to review your knowledge from last week. Do you remember those pairing rules? Which of those 4 nitrogen bases pair together every single time? Take a minute, pause the video, maybe review something from last week. If you need to go back in the video to see a list of those nitrogen bases, there's a list earlier in the video. I want you to tell me what bases pair together. All right, welcome back. If you need more time, feel free to pause the video, but we are going to move forward. So, to answer our question, remember we're thinking about those pairing rules. What bases pair together? Recall from last week, here's our answer. We always see adenine, or A, pairing with thymine. And we always see cytosine, or C, pairing with guanine, or G. These bases always pair together in these patterns. So as we add new nucleotides, and as we use or look at this parent strand as a template, we would be able to tell what nucleotides this DNA polymerase is adding each time. So, as I just got ahead of myself a little bit, we have an enzyme at work in step 2. Remember, step 1, That enzyme that was working, that protein molecule that was doing the work, was called helicase. For step 2, in base pairing, we have a different enzyme that's going to do work. That enzyme is called DNA polymerase. And it is the enzyme that is responsible for adding those new nucleotides to each strand. You'll notice on this image that there are 2 DNA polymerases working at a time. Remember, we had 2 parent strands exposed at once. Each of these parent strands gets nucleotides added to it. So each of these parent strands needs its own DNA polymerase. DNA polymerase only works in one direction, however. DNA polymerase can only add nucleotides to a template strand moving from a direction of 3' to 5'. This is really easy for one of the strands. Consider this top parent strand. It already goes in the 3' to 5' direction. This DNA polymerase can follow helicase as it unwinds the DNA. We call this strand that moves in the same direction as helicase the leading strand. It gets nucleotides added to it constantly, consistently. This template strand, however, isn't so lucky. We know that because these 2 DNA strands used to be joined together, if this one runs in a 3' to 5' direction, this one has to run in the opposite direction, meaning it runs from a 3' over here to a 5' over here. Which means that if DNA polymerase can only add nucleotides in the 3' to 5' direction, it is adding those nucleotides in the opposite direction that helicase is moving. So it is moving farther and farther away from the DNA that's being exposed as it's unwound and unzipped. This means that this strand of DNA lags behind. It gets its nucleotides added to it in short spurts, so we call it the lagging strand. So just to review the difference between that leading strand and the lagging strand. On the leading strand, our DNA polymerase is going to build that new strand in the same direction that helicase is unwinding in. They move in the same direction. Helicase is leading the DNA polymerase. So we call this the leading strand. It gets its nucleotides added consistently without any stops and starts. The lagging strand, however, gets built in the opposite direction from helicase. So helicase is moving one way, it's unwinding the DNA in this direction, and DNA polymerase is adding those nucleotides in the opposite direction, which means that this strand of DNA gets its nucleotides added in short spurts. So eventually, this DNA polymerase will keep adding nucleotides in this direction. Another DNA polymerase will need to come in and add nucleotides to this section. And by the time it does that, there'll be a new section that helicase will have already unwound that will need a DNA polymerase to add new nucleotides to. This gets a little bit tough to imagine, but just think about this lagging strand getting built in small pieces or fragments, rather than getting built as one long strand at a time. Leading strand and lagging strands get built in opposite directions. So that was step 2 of DNA replication. So remember, step 1, we unwound the DNA. Step 2, we added some nucleotides. Those 2 steps don't just happen at one single place in our DNA at a time. They happen at many places in our DNA at once. It'd be really, really inefficient if our cells could only synthesize new DNA at one place at a time. So we have developed the ability over thousands and millions of years to have this happen at many places at once. Here's an image to kind of help us visualize what those two steps look like. So we have our leading strand. Helicase over here would be unwinding in this direction. This leading strand gets replicated continuously as the DNA polymerase adds nucleotides one by one. This strand is the lagging strand. You'll notice that DNA polymerase can only add nucleotides in short bursts, which means that there ends up being these gaps in between stretches of nucleotides that have been added. There are gaps in that sugar phosphate backbone. This is why we have step 3. So step 3 is called joining, and it's all about fixing those small imperfections in the DNA molecule so that the strand is continuous. So it fixes those little blips where the sugar phosphate backbone hasn't been formed yet. These fragments on the lagging strand need to be joined in order to finish DNA replication. We call these fragments Okazaki fragments. They are named after a scientist with the last name Okazaki who discovered that these fragments existed and that the lagging strand was synthesized slightly differently than the leading strand. So to fix this issue where we have different fragments or Okazaki fragments of DNA on the lagging strand, we need an enzyme that's able to build that little connection on the sugar phosphate backbone. We call that enzyme DNA ligase. So DNA ligase is the enzyme that joins those Okazaki fragments by connecting their sugar phosphate backbones. So at this point, we've talked about all 3 of those enzymes. We've talked about helicase that unwinds the DNA. We've talked about DNA polymerase that adds those nucleotides. And we've talked about DNA ligase, which fixes those broken connections between sugar phosphate backbones. So what do we end up with once all of these steps are done? We've had our 3 steps. We've talked about our 3 enzymes. What is the result? So the result of DNA replication, as you might imagine, is 2 identical pieces of double-stranded DNA. So remember, we started out with a single piece of double-stranded DNA, 1 molecule of DNA. Through the process of replication, remember those 3 steps and those 3 enzymes, we now end up with 2 molecules of DNA that are identical to the original molecule. You'll notice on here we have our parent strand. It's still blue. In each of these new molecules of DNA, part of that new molecule came from the original DNA molecule. When we notice this, we can say that DNA replication is semi-conservative. It takes a little bit of the old and brings it in with the new. It semi-conserves the original piece of DNA. So that's the process of DNA replication. That's the portion of the video that you would watch or look over again if you wanted to figure out how to explain or show how DNA replication happens. This slide is all about that second learning goal, to being able to describe how DNA replication is different between different organisms. Specifically between eukaryotes and prokaryotes. So we just talked about DNA replication as being semi-conservative. That's true for both eukaryotes and prokaryotes. So regardless of the type of organism we always see semi-conservative DNA replication. We're always seeing a bit of that old DNA making its way into the new DNA. There are also several differences between DNA replication in eukaryotic organisms versus prokaryotic organisms. So eukaryotic organisms have linear DNA, which just means that our DNA is in long strands. So think about humans. Humans are eukaryotic organisms. We draw our DNA in our cells as long strands of DNA. Think about our chromosomes. They're long strands of DNA. If we wanted to synthesize our DNA, we could just start at one end and do it in one direction the entire way along our DNA strand, but that would take a really, really long time. Our bodies have developed something that's much better than doing it in one direction the whole way. We've developed the ability to have multiple replication points happening at once. So you'll notice on this portion of this eukaryotic organism's DNA, it has multiple origins for replication. So there's multiple points where helicase enzymes attached to this DNA and decided to unwind it, making those replication forks. This means that DNA replication can happen at each of these points in both of these directions. This allows our DNA to be copied relatively fast and efficiently. Prokaryotic organisms have a different structure for their DNA. Their DNA is often circular, which means that they would have one molecule of DNA that forms a circle or a plasmid within themselves, within the organism. So that DNA only requires one replication bubble. It's all connected end to end in a giant circle. So it will only have one origin of replication and two replication forks happening at a time. This doesn't necessarily mean that DNA replication takes longer in prokaryotic organisms. Often it goes just as fast, if not faster. If you're really interested in this, there are tons of great websites out there that can give you more information about these similarities and differences. I really encourage you to look more into that if you're interested.